Good morning. If you haven't noticed, our group uses a high contrast slide design. So my name is Chris Topper. I'm from Clemson University. My second year PhD student working in Dr. Marcus's group. And I'm just going to talk a little bit today about 2D LC for isolating and quantifying IgG and exosomes from cell culture media. So what are exosomes? They are a subset of extracellular vesicles, which are nanoscale membrane-bound particles. They typically range in size from 30 to 150 nanometers in diameter and contain phospholipid bilayer membranes enriched with tetraspanins such as CD9, 63, and 81, and these are what allow us to characterize them, and we'll talk about that a little bit more later. They're released by all cells, and they play a major role in intercellular communication, and they modulate homeostasis, promote tissue regeneration, and modulate the immune system. They contain DNA, RNA, proteins, and their cargo reflects the origin of the cell. And so, as such, they're being looked at to be used as biomarkers for detecting disease. But more recently, they're being looked at as vectors to deliver for targeted delivery of drugs to diseased cells, and they have low immunogenicity compared to lipid nanoparticles and adeno-associated viruses, and they can be used to deliver DNA, RNA, proteins, small molecules, and other biotherapeutics. And in fact, there's actually 2 phase 3 clinical trials underway. So monoclonal antibodies are manufactured proteins designed to recognize and bind to specific targets or the antigen, enabling precision medicine. And immunoglobulin G is the primary isotype used to make therapeutic mAbs. They modify the antigen binding site, and they can customize it to target many different targets. And so IgG is used for most antibody drug conjugates, enabling the targeted delivery of cytotoxic payloads to cancer cells, for example. Now, typically, mAbs are produced using Chinese hamster ovary cells cultured in bioreactors, and they're engineered to express IgG. And the IgG is then purified from cell culture media, typically using affinity chromatography, employing a protein A ligand for the initial purification at least. And the protein A has a high specificity for the IgG and utilizes the pH-based capture and elute mechanism. However, the ProA effluent is currently discarded as waste, and it is rich in exosomes. And so this presents an opportunity for byproduct valorization, and also at the very least, 2D online exosome quantitation, even a semi-quantitative, could provide an additional metric for process analytical technology when monitoring cell culture health and considering that exosome production is upregulated when in direct response to cellular stress. And additionally, offline analysis of exosome cargo might be correlated with process health states and prove even more informative. So the basic mechanism of protein chromatography involves binding at near neutral pH, and the protein A exhibits a high specificity for the Fc region of the monoclonal antibody, and elution occurs via a decrease in pH, which causes a conformational shift in the protein A, causing a partial unfolding due to altered electrostatics, and causes the mAb to dissociate. So our group employs capillary channel polymer fibers as the stationary phase. And these are just commodity fibers that are melt extruded with an 8-prong shape. They're non-porous to large biomolecules and exhibit excellent mass transfer properties in viscous fluids, such as cell culture supernatant. They're cheap, costing about $5 per column, and column chemistries can be modified on demand. One of these we use is polypropylene, which possesses significant hydrophobicity, and we exploit this to create our protein A modified columns, which is essentially we take a solution of recombinant protein A, flow it through the column, the fiber surface is covered in the protein A, and it allows us to bind about 2 IgG per molecule of protein A. The other common fiber that we use is polyester. and this is moderately hydrophobic, and we've used this for hydrophobic interaction chromatography, reverse phase, used them as the desalting columns and as capture loops. And so when packed into a column, the fibers, the 8-prong shape, interdigitate, forming hundreds of micrometer-sized capillary channels. We'll zoom in picture. And so our group has used hydrophobic interaction chromatography on these PET fibers to isolate exosomes previously. The sample is loaded under high cosmotropic salt conditions, typically 2 molar ammonium sulfate, and the polar and ionic species pass through unretained, while proteins and exosomes adsorb on the fiber surface. And then the salt concentration is decreased, causing compounds to elute based on hydrophobicity. And for very large or very hydrophobic compounds, sometimes a little organic modifier is required to facilitate desorption. And so we sought out to develop a ProA HIC method that employs HIC in the 2D to isolate exosomes from 1D ProA waste streams. And the idea of implementing this downstream was to possibly mitigate the GMP regulatory burden on companies who might be throwing away these exosomes in their ProA effluent. The challenge, however, is the 1D effluent results in exosome breakthrough in the second dimension. And so we need sufficient salt concentration to get the exosomes to stick. And so the problem is an at-column dilution, and I say dilution in quotes because we actually need to increase the salt concentration prior to hitting the second dimension column. So this is a little schematic of the instrument configuration. We used Agilent Infinity 2 LCs consisting of a pump, a binary pump, a ProA column in the first dimension, and this can be our CCP fibers or any commercial column, a UV detector for IgG quantitation, a modulator, which was simply a two-position 8-port switching valve, a second binary pump, a mixer, which was a 100 microliter microfluidic jet weaver mixer. And this was just to ensure, because the starting mobile phase in the second dimension is 1 molar ammonium sulfate, we added this mixer just to make sure that there was adequate mixing and also to maybe reduce any refractive index artifacts in detection. And so the second dimension column consisted of our PET CCP fibers, followed by a detector for exosome identification and quantitation. And this allows us to collect the IgG and both the exosomes. Okay, so the modulation strategy that we employed, we'll call the light green salt sugars, the IgG in the middle here, and the exosomes in pink. And so as the sample is loaded and the supernatant passes through the ProA column, the IgG is adsorbed while the effluent begins to fill the first sample loop. Have the 1D effluent in red and the 2D in blue. And there's a piece of blue in the first sample loop for a reason. The idea is we used two 200 microliter sample loops and only partially filled the sample loop to help aid in dilution of the 1D effluent before reaching the second dimension column. So when about partially full, the valve switches while the 1D effluent begins filling up the second transfer loop, and the 2D pump pushes out the 1D effluent from the initial loop to the mixer, flows through the HIC column, where the exosomes adsorb and all other constituents of the supernatant pass through to waste. And this switching process continues until all the supernatant has been loaded through the ProA column. The columns are then flushed, and then compounds of interest eluted. There's a little chromatogram of the 1D separation, so absorbance on the y-axis, time on the x. And so this gray box is the transfer window. So during this transfer window, the flow rate of the 1D pump was 0.1 mils per minute, while the flow rate in the second dimension was kept constant at 0.5. And so this was just due to our limitations, not having active solvent modulation, not having 2D software, having to program all this manually, and just to facilitate the timing of things. And so as those sample loops would partially fill, they would transfer over. Sorry, I'll back up here. So all the supernatant response, the flow through, fractions are being transferred over at this time, and then mobile phase is switched to citric acid, causing a decrease in pH, causing that partial unfolding of the protein A and dissociation of the IgG. What our 2D HIC chromatogram looks like, there's multiple peaks here. That just signifies the various valve switches and transfers to the second dimension. That's all supernatant flow through from here. And so the supernatant transferred over, this is all one unretained 1D effluent with 1 molar ammonium sulfate and 15% ACN. And this allows only the exosomes to bind, while host cell proteins, salt, sugars all pass through unretained. To elute the EVs, the salt concentration is decreased to 0 with 40% acetonitrile and PBS added as a modifier to help peel them off. And so to evaluate modulation efficiency, we ran HIC in the first dimension, looked at peak areas of an exosome standard, and measured them in the absence of any modulation. We just wanted to see if all the mass was being transferred over to the second dimension. And then we ran HIC in the 2D, looked at exosome peak area, compared that relative to those in the 1D without any modulation. And so we looked at 2 different modulation frequencies, 60 seconds and 30 seconds. And so with 60 seconds, the sample loop is filled, 50% of the sample loop volume is filled with 1D effluent. Whereas with the 30-second frequency, only about 25% of that 200 microliter sample loop is filled with 1D effluent, and the remaining is of the 2D mobile phase. And so you can see when the frequency was 60 seconds, there was not enough dilution or increase in salt concentration in that sample loop for exosomes to bind effectively. However, with 30-second modulation valve switches, we got essentially 95% of the peak area measured with just 1D HIC. And so looking at IgG recovery, CHO cell supernatant from a K1 cell line was spiked with various concentrations of IgG, and the K1 cell line does not express IgG, and so we were able to then look at recoveries and directly compare spike peak areas to no column injections of neat IgG standard. And so our CCP columns and adjuvant commercial ProA column performed comparably. Quantitative recoveries were seen with relative standard deviations of less than 5%. I believe all these recoveries for the CCPs were between 92 and 98%, and that of the adjuvant between 94 and 105%. To assess the purity of these fractions, SDS page was performed. The samples were denatured using dithiothreatol, denatured by boiling at 95C, and then loaded on a 12% polyacrylamide gel. You have molecular weight ladders here in 1 and 10, an IgG standard loaded in lane 2. These 2 bands correspond to the 50 kilodalton heavy chain and the 25 kilodalton light chain, respectively. Lane 3 contains the recombinant protein A that we use to make our columns, to modify our CCP columns, and we loaded that just to ensure that nothing was leaching off of the ProA CCP column. Lane 5 is IgG fractions collected using our CCP column. These are pooled triplicate fractions with blank runs run in between each, and those were pooled and loaded in lane 4 just to see if there was any carryover or any ProA leaching. The same thing for lanes 6 and 7 for the commercial column, respectively. Lane 8 was left blank as it neighbored the supernatant, which is messy and contains a ton of host cell proteins. And so looking at this, for both the commercial and our modified ProA columns, we just see nice bands at 50 kilodaltons and 25, and no carryover or ProA leaching in either column. And we also plotted the log molecular weight versus relative migration distance of the bands, and the calculated molecular weights of the bands were in good agreement with the IgG standard. To characterize the exosomes, we used nanoflow cytometry, and the particle size distribution had an average mean diameter of 83 nanometers, and so this is well within the range of typical exosomes. We also used immunolabeled antibodies for anti-CD9, 63, and 81, and these were to identify actual exosomes rather than just other microvesicles or apoptotic bodies. And then we used a memglow, which is a lipophilic anchor fluorescent tag, and that essentially binds to any kind of vesicle particle by embedding itself in the membrane, and that signal is shown on the x-axis, while the tetraspanin fluorescence is shown on the y. And this red quadrant up here is what we want to see for exosomes. This linear trend shows that as vesicle size increases, so does tetraspanin fluorescence as there's more tetraspanin proteins on that membrane. And this bottom quadrant here is just memglow labeled vesicles without any tetraspanins. So about 66% of the particles we believe to be exosomes, and of that fraction collected from the 2D, 93% of those were indeed vesicles. To determine exosome recovery and purity, we generated a response curve by injecting serial dilutions of CHO-derived exosome standard, and determined that in the 2D fraction, we had about 1.76 times 10 to the 11 particles per milliliter, with a relative standard deviation between triplicates of less than 7%. We then ran a micro-BCA to determine protein concentration in these, and saw a greater than 99% reduction in host cell proteins from the supernatant to the collected exosome fraction. The CHO supernatant had about 4 milligrams per milliliter, while the exosome fraction had about 3. And so this is what the NIH uses to determine exosome purity by reporting particles per microgram protein. And our separation came in at around 5.86 times 10 to the 10th particles per microgram protein. And this meets the NIH guidelines for high purity exosomes, determined to be 3 times 10 to the 10th particles per microgram. So currently our group is working on scaling up our CCP fiber columns to 2.1 millimeter ID analytical size. We're also doing ytterbium labeling of exosomes for quantification using ICP-MS, and performing cellular uptake studies of the HIC isolated exosomes to evaluate if the acetonitrile impairs physiological functioning at all. For myself, currently I'm working on anion exchange separations using polyethylene imine modified fibers, and the idea of this would be to generate a different modality to isolate exosomes without having the need for organic modifiers. Looking at other novel fiber modifications to access new separation modalities, and then we'll be looking at separations of liposomes and adeno-associated viruses. So, in summary, we successfully demonstrated that the use of a 2D LC to isolate and quantify both IgG and exosomes from supernatant shows the possibility to extract value from current waste streams and possibly expand process analytical technology to monitor cell culture health and improve QA/QC. The low-cost ProA CCP fiber columns did achieve product purity comparable to leading commercial columns. No carryover IgG or ProA leaching was observed. And we did successfully isolate high purity exosomes downstream of the ProA separation, seeing a greater than 99% reduction in host cell proteins from the supernatant and achieving a product purity of greater than 5 times 10 to the 10 particles per microgram. I just want to thank the committee for inviting me, the NSF for funding this research, Dr. Marcus, and my group members. Thank you, and I'd love to answer any questions.
So unfortunately, we're a little behind schedule, but there's only one talk between you and lunch, so I think be plenty of time to talk during lunch. But thank you for a fascinating talk. So our last speaker before lunch. Is Dr. Matt Sorensen from Eli Lilly and Company. So we're coming back to the pharmaceutical industry and I think talking about still different molecules.
Yes. Thank you. All right, thank you. It's an honor to be here. First time attendee. It's been a great day and a half so far. Looking forward to the rest of the week. Like Dwight said, I'm the last person standing between lunch, so I'll try to make it fast, yet still exciting. Speed and quality, like Jane said, as a pharmaceutical industry. Bulk of my talk is on 2DLC of oligonucleotide therapeutics. So I want to briefly introduce oligonucleotides as therapeutics. There's a broad spectrum of oligonucleotides. The short version is oligos. They are polymers of nucleic acid building blocks, right? The same building blocks in DNA and RNA in our own body, A, G, C, T, and U, so adenosine, guanine, cytosine, thymidine, and uracil nucleic acids. A big advantage of oligonucleotides as medicines or as therapeutics is that they can be highly targeted. There's a number of different oligonucleotide therapeutics. mRNA vaccines are obviously really big these days. siRNA or siRNA therapeutics is what I'll be talking about in my talk. ASOs, antisense oligonucleotides. So it's a broad spectrum of oligonucleotide therapeutics. And a big advantage of oligonucleotides is that they can be highly specific. They oftentimes target specific genes in the body and either knock down those genes that cause disease or do some other highly specific modulation of different genetic molecules in our body. Oligonucleotide therapeutics specifically are heavily modified. So if you were to just inject DNA or RNA from something you make or from some other organism, our body will degrade that easily. It'll recognize it as a foreign entity. And so as a therapeutic modality, we want to modify slightly so the body doesn't degrade it and so it can actually perform its therapeutic effect. So I'll highlight some of the modifications because they're important from an analytical perspective. This slide on the top here demonstrates some of those modifications. So the ribose sugar can be modified on the 2' position. So in DNA, it's just a hydrogen, and RNA, it's an OH group. In therapeutic oligonucleotides, it's usually a fluorine or an O-methyl group that really increases the bioavailability. They're oftentimes modified or conjugated to lipids or peptides or sugars to increase their targeting effect to certain organs in the body. And that's usually on the terminus here, 3' or 5' end. And then another important modification is on the phosphate backbone. A non-bridging oxygen is substituted for a sulfur group here to make a phosphorothioate bond. This complicates, so the advantage from a therapeutic perspective is that this really increases the bioavailability and decreases the nuclease activity in vivo. So your body won't just degrade the medicine right away and it can actually do its therapeutic activity. From an analytical perspective, though, this creates a diastereomer, which I'll talk about later in terms of when we want to separate the diastereomers, when we don't want to separate the diastereomers so we can separate the other impurities in our drug substance or drug product. So my talk will focus on a number of different 2DLC implementations that we've done at Lilly to help guide process development and improve our impurity characterization of different siRNA drug substance and drug products. This includes size exclusion, anion exchange, ion pair reverse phase or IPRP, and hydrophilic interaction LC or HILIC. So the first case study I want to talk about is when we were doing process development in our development labs where we're trying to optimize our synthetic route. We have a number of different synthetic approaches that we use, and then on the analytical side we can guide the quality of those synthetic approaches. And so one assay for siRNA is that we do a non-denaturing duplex purity analysis. So siRNA is a double-stranded nucleotide or oligonucleotide, similar to our DNA in our own body. It's double-stranded, right, with the Watson-Crick base pairing by hydrogen bonding of the two strands. So synthetically, we make the two strands separately, and then we combine them in a 1-to-1 ratio. They'll hydrogen bond under aqueous conditions. And one of the analytical assays we run is this non-denaturing purity analysis to know we roughly have a 1-to-1 stoichiometric ratio of our antisense and our sense strand in this duplex drug substance. But one of the synthetic approaches that our chemists were implementing, a new impurity showed up, right? So we want to characterize this new impurity. The non-denaturing assay is a size exclusion assay with PBS mobile phase, so it's not very mass spec friendly. And so a 2DLC approach was one of our first approaches to tackle this new impurity. Obviously, we can do spiking studies with known compounds to make sure it's not something we already know. So I spiked an extra antisense, which is a single strand. One of the two single strands is in the duplex. Spiked in the sense strand, and neither of these were our unknown peak. So we used 2DLC with a mass spec compatible ion pair reverse phase modality in the second dimension to try to characterize this unknown peak that's in the high molecular rate region of our SEC chromatogram. So for the 2D LCMS experiment, we cut all 5 peaks, or we analyzed all 5 fractions in a multiple heart cut mode with the conditions there. So the second dimension conditions are, again, ion pair reverse phase, where it's a C18 column at high temperature to denature any secondary interactions. And then the mobile phase is a diisopropyl ethylamine ion pairing reagent, which increases retention of our oligonucleotide on the C18 column, and its hexafluoroisopropanol modifier really increases the mass spectrometry compatibility. And we're able to get good retention, good separation of impurities within each cut, and we also were able to get high resolution mass spec data with coupling online with a QTOF, and this really informed what our impurity was. I'm not going to go into detail on what it was, but it really informed our process chemists in a pretty timely manner, right? As Chad was pointing out, we don't have to fraction collect or send it to our fractionation or purification group. We can rapidly give our process chemists some information that they can then modify, or we better understand what the impurity profile is with these different synthetic approaches. The second case study I'll talk about is an anion exchange coupled with ion pairing reverse phase, 2-dimensional LC approach for antibody RNA conjugates. So again, Chad gave a good background on ADCs. Antibody RNA conjugates is a subclass of ADCs where the drug is an RNA, and the antibody is the carrier to try to target the RNA to a specific organ in the body. And so a similar chromatogram is shown here for the drug-antibody ratio using AEX. But again, the high salt concentration makes this very not mass spec friendly, and we're not really able to get good analytical data on our RNA or our oligonucleotide. And so the second dimension, again, was an ion pair reverse phase denaturing assay, where it was really informative to know the quality, in particular, of the excess siRNA that didn't get conjugated, whether or not we could forward process or purify, rather, the excess siRNA that did not get conjugated. Can we reuse that, or is it degraded? So it's very informative, again, to our process chemists. The next few slides then are more of a deep dive on purity and impurity analysis for oligonucleotides. So as Jane pointed out, impurities are of great concern, and we have a lot of eyes on what the impurity profile are for our drug substance and drug product molecules. So for oligonucleotides, ion pairing reverse phase, or IPRP, is the gold standard for impurity profiling. It's able to resolve a lot of impurities of both single-strand and double-stranded siRNA duplexes. The common impurities are truncation products, so these are synthetically made, and so each addition of that nucleotide is not always 100% successful, and so you'll get N-1, N-2, N-3, and that's what you see here with the chromatographic profile. And sometimes you'll get a double addition, so you see some small peaks on that are retained higher, and so you can see good resolution. Depending on your ion pairing reagent, you can optimize the resolution. But obviously, it's a complex molecule, and you still get coelution around your main API peak, and so we really want to try to leverage some more separation techniques, namely 2DLC, to try to better resolve some of the closely related impurities while maintaining separation of the broad range in impurities we see with these synthetic crude oligonucleotide drug substances. So our approach first was to just couple ion pair reverse phase in both dimensions using the same mobile phase conditions and the same column, but drastically different gradient separations. So the first dimension was used to broadly separate a wide range in polarity of our truncations and other impurities. So you'll see pretty good utilization of the separation space, but this was a fairly steep gradient in terms of the oligonucleotide, a 3 to 15% B gradient over about 20 minutes, which is pretty steep for oligos. Oligos are very sensitive to changes in the percent B, and so that's a pretty steep gradient effectively. And so our second dimension is we use multiple heart cutting to cut the API peak, and then do a very narrow second dimension separation, again, just changing the gradient range or the gradient slope. So a very shallow slope in the second dimension of about 2% B over 10 minutes. And this really pulled out some impurities. It's mass spec compatible, and so we can get better resolution with this very narrow gradient slope, and we can also still get mass spec data to inform what these impurities are. Of course, there's still peak overlap, and so our next move was to use a more orthogonal separation approach, which was HILIC. And so in a similar vein, we do ion pair reverse phase separation in the first dimension. We can separate N-1, N-2 oxidation impurities, defluorination impurities, but you still see peak overlap. And so HILIC has some unique selectivity and orthogonality differences compared to ion pair reverse phase for oligonucleotides, and so we really leverage that selectivity difference. So if you just do high-res mass spec on some of these peaks, you can see very different or very clear overlap of N-1s and then our oxidation impurities. And previous work, we looked a lot at selectivity differences between ion pair reverse phase and HILIC, and we know that the oxidation impurities are more hydrophilic, and so they get retained more under HILIC conditions rather than ion pair reverse phase conditions. And so it was a perfect coupling of ion pair reverse phase with HILIC to get a better separation of these N-1 and PO impurities. And so this is what is shown here, where we're in both cases we have coelution of N-1s, different N-1s, so adenosine versus uracil, and then oxidation, so that phosphorothioate species being oxidized to a phosphodiester bond. A lot better resolution with HILIC in the second dimension than with ion pair reverse phase in the first dimension. So the final application then, and it's still work in progress here, is what we're trying to do is couple our impurity method in the first dimension with a diastereomer separation in the second dimension. So as I mentioned in the intro here, diastereomers are present whenever you have this phosphorothioate linkage, these PS bonds. They're very common in therapeutic oligonucleotides because they increase the resistance of DNA or RNA to nuclease activity in vivo. So if you inject a non-modified or at least no PS bonds in the body, it'll get degraded right away. If you add some thiol bonds on the terminus of your sense and antisense strands, the in vivo activity is greatly increased. And so profiling the diastereomers is important from a safety perspective, right, and a process control perspective to say, okay, our process is consistently making the same diastereomeric profile, and we're confident in our process. So analytically, we need to be able to separate the diastereomers and say, yep, it's the same profile batch to batch from a QC perspective. So there's a couple of different advantages of applying 2DLC for both purity and diastereomer separation. One is that it can speed up our analysis with this multi-attribute method. The second is that oftentimes, or there are certain synthetic approaches that we're not able to have our sense and antisense strands separately. There are certain convergent synthetic routes that only make a duplex, and so we need to denature that duplex and then do diastereomer profiling in the same assay. And that's the goal of this work, which is still ongoing, but I'll share some initial learnings in the next slide or two. So the chromatogram shown here is a denaturing purity assay. Because the antisense and the sense strand are just hydrogen bond Watson-Crick base pairing, high temperatures denature that, and we're able to get good resolution of key impurities, down to the low percentage to 0.05 or 0.1% impurities. The goal then is in the second dimension to resolve the diastereomers. So for our antisense strand here, there's in theory 32 diastereomers. So for every PS bond, it's 2 to the n number of PS bonds. So there's 5 PS bonds, so 32 diastereomers total. Our sense strand has 1 PS bond, so that's only 2 diastereomers. You'll see the chromatograms look like garbage in both cuts, so this is not what we expect. For reference, the diastereomer method is shown here in gray, where it's 1D only. So something's going on here that we need to figure out. Why is the separation not working in the second dimension? So the conditions are shown on the left-hand side. The first dimension is, so they're both ion pairing reverse phase methods, but the difference here now, instead of the slides a few slides ago, is that the ion pairing reagents are different. So the modifiers are different. So in the first dimensions, it's DiPA, diisopropylethylamine, and HFIP, hexafluoroisopropanol. Gives you really good purity resolution of the duplex, the denatured duplex. And then the second dimension is triethylamine with acetic acid as the acid modifier, so TEAA. And so we think that the DiPA and the HFIP are still getting retained and influencing the separation condition in the second dimension, such that it's not performing as we would expect in the 1D only reference. So if we switch to TEA in both dimensions, it looks a little better, but again, we still have HFIP in the first dimension, which is necessary for the chromatographic resolution we obtain, but the antisense and sense strand are starting to look like what we expect. So antisense, we typically see about 12 peaks. In the sense strand, we usually see 2. You can kind of see 2 blobs, and then what's looking like 12 blobs, but still not what we expect from what we got from the first dimension. And so I started to do some basics of, okay, let's just do a pseudo 1D where we inject just without a column, and we have pure water acetonitrile. We're doing active solvent modulation to focus our oligo on the head of the column in the second dimension. This is the sense strand where we see 2 peaks. Okay, so everything's looking good if you just have water and acetonitrile. So we know we have a reasonable suspicion to think that the ion pairing reagents are causing issue in the second dimension. And so we've begun to then characterize the retention of our DiPA and our HFIP in the second dimension that's coming from our first dimension effluent, and know, okay, it's sticking around for, 5, 10, 15 minutes, depending on our percent B in the second dimension. And so knowing this, then we can say, okay, let's hold off executing our second dimension gradient until all of our first dimension ion pair and acid modifiers off. and then we can execute our second dimension diastereomer gradient and separate our diastereomers. So we know, again, that oligonucleotides are very sensitive to our percent acetonitrile. So as long as we operate in this, sub 5% regime for, 5-10 minutes, our DIPEA, our HFIP will elute eventually. Our oligo will just stick to the column for days and days at sub, 6-7% acetonitrile. So we're looking forward to then trying to pursue this further, knowing what we know now about the retention mechanisms of our ion pairing reagents and of our oligonucleotide. So with that, I hope I just provided a quick, snapshot of some 2DLC implementations in our process development labs at Lilly, focused on oligonucleotide therapeutics. It's a really great team to work with at Lilly across the board. The group is shown here along with our management support, which has been phenomenal for our capability builds at Lilly. With that, happy to take any questions at lunch. So thank you, Dwight, and thank you to the organization.